I'm just going to cover passive transport in this lecture, and we'll do osmosis and the rest of these in some following lectures. So we're just going to look at simple diffusion, facilitated diffusion, and diffusion through channels in this part. And remember, passive transport, we're going to be moving from high concentration to low concentration. So we're going to go down the gradient, and we don't need any energy to do this. In simple diffusion, molecules are just going to diffuse right through the cell membrane, and there's no transport protein that's needed. And remember that diffusion, that is a form of passive transport. So anytime you see the word diffusion, that means passive. So molecules are going to move down the concentration gradient from high concentration to low concentration, and we don't need any energy to do this. The types of substances that are transported by simple diffusion are hydrophobic nonpolar molecules and also small polar molecules like water. So some examples here, some hydrophobic nonpolar molecules would be things like oxygen and carbon dioxide. We could have some of our lipids, like fatty acids. And then also we have some fat-soluble vitamins. Those would be vitamins A, D, E, and K. You don't have to memorize that. We'll talk about that when we get to the digestive system. We'll come back to that. But those are hydrophobic. And then the other one would be water. Okay. So remember, water, even though it is hydrophilic, because it's so small, it can slip through the gaps in the phospholipid bilayer as the phospholipids are moving around. In simple diffusion, molecules are going to move from high concentration to low concentration, and the molecules will move across until they reach equilibrium, and they're the same on both sides. There are several things that affect the rate of simple diffusion. So we have the size of the chemical driving force, the membrane surface area, and the membrane permeability. So if we look at the size of the concentration gradient, the larger the concentration gradient is, the more molecules you're going to move across the membrane. So for example, up here on the top left, we have a concentration gradient. We have 1 molar on the left side and then 0 on the other side. And so our chemical driving force is going to be pretty big. So we've got a pretty thick arrow there. And then over here on the right, we have a smaller concentration gradient going from 0.75 molar to 0.25 molar. And so you can see with a smaller concentration gradient, we're not going to be moving as many molecules across. And so our chemical driving force, our arrow there, will be not as thick, so it's not as strong. And then down here on the bottom, we have the molecules at equilibrium. So we've got equal concentration on both sides. So there's going to be no net movement of the molecules from one side to the other. And then this is just showing the effect of the size of the concentration gradient on the net flux. And net flux is just talking about how many molecules are moving across the membrane. So you can see that as the concentration gradient gets larger, you can move more molecules across. Membrane surface area also affects the rate of simple diffusion. And if we have a membrane with a large surface area, like on the left, versus a smaller surface area on the right, the larger the surface area, the more that we're going to be able to get more molecules across, because you've got a larger surface area to do that than if you had a smaller surface area. And so that's why the lungs have a huge surface area to do gas exchange. If you were to take the lungs and flatten them out with all the alveoli, the air sacs in there, the surface area of the lungs is about the size of a tennis court, which is really big. Other things that affect the rate of simple diffusion would be some membrane permeability factors, how hydrophobic the molecule is. So the more hydrophobic it is, the easier it's going to be able to get across the membrane. Smaller molecules, whether they're hydrophilic or hydrophobic, will cross faster. Temperature. The higher the temperature, the faster the molecules move, and then you're going to move more molecules across the membrane. But remember that the body doesn't really change the body temperature that often unless maybe you have a fever, because we try to keep that in homeostasis. And then the thickness of the membrane is also important. So the thinner the membrane is, the more molecules that we can get across. And we're going to see that again when we get to the respiratory system, because the alveoli, when we do gas exchange there, going through the alveolar wall and then the epithelial layer of the blood vessel, that's only 2 cell layers thick. So it's very, very thin. So you're going to be able to get a lot more gas exchange and get more molecules across. And that's why we don't have gas exchange, for example, in the trachea, because that tissue is just too thick and we're not going to be able to diffuse molecules across there. Facilitated diffusion is another form of passive transport. And remember, diffusion, anytime you see that word, you know it's got to be passive. So molecules are going to be going down the concentration gradient and there's no energy required. Facilitate means to help, and we're going to be using a carrier protein to help hydrophilic molecules across the membrane. And we're going to be moving things like larger polar molecules, for example, glucose and amino acids. They'll be moved across by a carrier protein. We also have ions in water, which are also hydrophilic, but we're not going to be using a carrier protein for them. We're going to be using a channel protein, which we'll talk about next. And the carrier protein is going to change shape as it binds to the specific molecule that it's going to transport across, and then it will flip it to the other side. So you can always recognize a carrier protein because it's going to change shape like that. In this example, we've got a higher concentration of our molecule inside the cell than we do outside. And so you can see that we are definitely going down the gradient, moving from high to low concentration. So no energy is needed to change the shape of the carrier protein. It just does that on its own. The carrier proteins can move the molecules in both directions, but they're not going to be transporting this molecule from outside the cell to the inside just because of the concentration gradient. You've got a lot more of the molecules on the inside, more of a chance of it binding on the inside than you do on the outside because there's a lot fewer molecules there. But they could reverse direction and go the other way if the concentration gradient did change. Here's just an example of facilitated diffusion moving glucose. So you can see that looking at our concentration gradient, we've got a higher concentration outside the cell than inside the cell. And you can see our carrier protein, very specific. It will only bind to glucose. It's not going to transport anything else. And then when it binds to the glucose, that causes it to change shape and it flips it to the inside of the cell down the concentration gradient. So again, no energy needed for this. This is passive. There are a number of factors that affect the rate of facilitated diffusion. Each carrier protein just has a specific speed that it works, and some work faster than others. Also, the number of carrier proteins in the membrane is important, and then the size of the concentration gradient. And the thing that's really important to note about facilitated diffusion is that saturation can occur when you get a high concentration gradient. So this is just showing you saturation. So with our concentration gradient, we can move more molecules across the membrane the higher the concentration gradient gets. But at a particular point here, our graph levels off. And even though the concentration gradient gets higher and higher, you get more molecules building up on one side of the membrane, you can't get any more molecules across any faster. And that's because you only have a certain number of carrier proteins in the membrane. And so the carrier proteins can become saturated. So that would fill in our blank up there. Carrier proteins become saturated. Okay, and we don't see that in simple diffusion. So simple diffusion, that line would not level off like this. It would just keep going straight up. Okay, because in simple diffusion, you're not using any type of transport protein, so there's nothing to become saturated. If you want the carrier proteins to become unsaturated, you'd have to put out more carrier proteins in the membrane to get the line to go up again. And then eventually it would become saturated at a higher point, and then you'd have to put more carrier proteins out. So the cell could certainly change the amount of carrier proteins that are in the membrane. Another passive form of transport is diffusion through channels. And we're going to be using a channel protein which has a hole or a pore in it. And because we're using the word diffusion, we know it's going to be passive. So we're going to be moving down our concentration gradient and no energy is required. And each of these channel proteins have a specific shape and they're only going to be transporting a specific molecule across. So we're going to be using these channels to transport water and ions across the membrane. So both of these are hydrophilic. They're not able to cross on their own except for water. Water can do simple diffusion through the membrane, but it's pretty slow. So if you want to move water across the membrane quickly, it's much faster to use these aquaporins, these channels for water. The ion channels, we have two different types. There's leak channels, which are open all the time, so they're always leaking. The ion is always moving through there. And then we have ion channels that have a gate on them. So there's some different things that can trigger the gates to open or close. Some of the gates are ligand-gated and other ones are voltage-gated. So when the gates close, the molecule can't move through, but if it's open, then the ion can go through. Here's a picture of an aquaporin on the left. So this will be used to transport water across the membrane. And this is going to be much faster than the water doing simple diffusion without using the channel protein on its own. And we also have an ion leak channel on the right. And these are open all the time. There's no gate on these. And so they're always leaking. Our gated ion channels, different things will open and close the gates. So with a voltage-gated ion channel over here on the left, our gate here that's in blue, that's going to be triggered to open when the membrane potential, the inside of the cell, becomes less negative and more positive. And we're going to be talking a lot about voltage-gated ion channels when we get to the neurons in the muscles. And then we also have ligand-gated ion channels. Remember, a ligand is just a very general term. It just means something that's going to bind to a protein reversibly. And these ion channels are made out of protein. And in this case, our ligand is the neurotransmitter acetylcholine. Okay, and that's going to be released by the motor neuron. And maybe we're looking at a skeletal muscle here. And the gate in the middle will be opened when our ligand, our acetylcholine, binds. And that'll open the gate, and this one happens to let sodium ions in, and then that will trigger contraction of the skeletal muscle. And then how we get the gate to close is when the acetylcholine falls off of our ligand-gated ion channel there, and then that will close the gate. There's a couple of factors that affect the rate of transport for ion channels as well. Each ion channel has a specific transport rate. The number of channels in the membrane is important, and then whether the ion channels are open or closed if they're a gated channel. Ion channels can get saturated, but it's much easier to saturate the carrier proteins than the ion channels. And then this is just a reminder of the difference between the channel proteins and the carrier proteins. So the channel proteins just have a channel or hole in them. They're not going to change shape. They're not going to flip back and forth. Whereas the carrier proteins do change shape. So they're going to flip back and forth to move the molecules across.